Every second, inside every cell in your body, molecular chains are rapidly twisting and locking into highly complex, three-dimensional structures. Before a protein takes its final shape, it starts out as a simple, one-dimensional chain of amino acids. The specific sequence of those amino acids acts as a strict chemical code. That exact order is the sole factor determining how the entire protein will fold. Once that chain folds into its final three-dimensional geometry, that physical shape dictates the protein's operational function within the cell. Determining this exact structure is a medical necessity. When proteins misfold into incorrect geometries, the resulting mechanical failures are the root mechanisms behind diseases like Alzheimer's, Parkinson's, and cystic fibrosis. Historically, mapping these three-dimensional structures required scientists to isolate the proteins in physical laboratories to measure them directly. One primary method is X-ray crystallography, which involves bouncing radiation beams off crystallized proteins to trace their internal atomic arrangements. Another is nuclear magnetic resonance, or NMR spectroscopy, which maps the proximity of atoms by measuring their magnetic properties. A third major technique is cryo-electron microscopy, which freezes proteins and fires electrons through them to build a structural composite. All three of these physical methods are highly accurate, but they also require massive financial investments, highly sensitive equipment, and months of manual labor to map a single protein. DeepMind's artificial intelligence system, AlphaFold, bypasses the laboratory entirely. It shifts the challenge of determining a protein's structure from a physical measurement bottleneck into a purely computational prediction problem. At its core, AlphaFold is a deep neural network built specifically for complex pattern recognition. To build it, DeepMind researchers fed the network a massive dataset containing millions of known amino acid sequences alongside their corresponding experimentally verified 3D structures. The system learned using a standard machine learning paradigm known as supervised learning. In this training loop, the model generates a prediction for a given sequence, compares its prediction against the known ground truth structure in the dataset, and adjusts its own internal parameters. The primary objective of this loop is to continually minimize the mathematical delta between the shape it computes and the actual physical reality. By running millions of these iterative cycles, the model implicitly deduces the complex physics and chemical rules of protein folding directly from the raw data. To see how this works, we can look at a schematic of the AlphaFold computational pipeline. On the left side, we start with the raw input, a single, unmapped, one-dimensional amino acid sequence. But the system does not process this sequence in isolation. First, it generates a multiple sequence alignment, stacking the input against a grid of similar sequences from other organisms. This allows the model to scan for evolutionary relationships. By checking how the protein varies across species, it flags matching evolutionary traits. This data then passes into the first major architectural module, the evoformer. Inside, the evoformer overlaps the sequence alignment data with a separate two-dimensional matrix representing pairwise interactions. The mathematical goal here is precise. It correlates the data to identify which specific amino acids are evolutionarily linked, meaning they are highly likely to sit physically adjacent to each other in 3D space. By the end of this stage, the evoformer has successfully translated raw evolutionary history into a highly probable map of spatial proximities. This pipeline then expands as the correlated data passes into the second half of the architecture, the structure module. This module's function is to apply strict spatial constraints to the proximity data, translating those data points into a rudimentary 3D wireframe. Through a process of iterative refinement, the model adjusts the angles and distances, locking the exact physical geometry of the protein into a stable configuration. This continuous pipeline, processing a 1D sequence input straight into a 3D structural output, is known as end-to-end learning. Operating end-to-end means the model executes this entire workflow without requiring any intermediate steps or manual feature extraction from human scientists. The architecture bridges the gap between biological abstraction and exact physical coordinates entirely on its own. Validation of this architecture is clear in the CASP competition graph. The x-axis tracks years, the y-axis plots predictive accuracy. After decades of incremental progress, in 2020, AlphaFold delivered a massive leap, defeating legacy models by achieving atomic-level accuracy. This computational speed immediately accelerates pharmaceutical drug discovery, allowing researchers to map disease mechanisms and identify therapeutic targets in days rather than decades. It also provides immediate utility in modeling complex biological processes, like enzyme catalysis and rapid protein-protein interactions. By publishing these predictions in open-access databases, the system removes the requirement for specialized laboratory hardware, allowing researchers to initiate drug discovery projects in hours rather than months. Yet, despite its success in the CASP competition, the architecture is not infallible. First, running AlphaFold requires immense computational power to both train the neural network and process inference queries, creating a strict resource barrier for many researchers. Second, the model struggles to accurately predict the structures of highly flexible, intrinsically disordered proteins that lack a single stable state. And third, it suffers from the black box nature inherent to deep neural networks. This lack of interpretability means that while researchers receive an accurate prediction, they cannot fully trace the exact deductive steps the model took to finalize that specific spatial geometry. Still, the success of the evoformer and structure module is currently spurring the development of specialized AI models designed to map protein-ligand interactions and model broader systems biology. The AlphaFold architecture serves as a functional map, translating the raw chemical code of amino acids into the foundational mechanical rules of molecular life.